హలో ఎవ్రీవన్, ఎలా ఉన్నారు? సో ఇవాళ నుంచి బయాలజీ స్ట్రెస్ పోయినట్టే కదా. సో హౌ వాజ్ యువర్ ఎగ్జామ్ ఎలా రాశారు? ఎవ్రీవన్, ఎనీవన్ హ్యాస్ ఎనీ డౌట్? సో వాట్ డూ యూ థింక్, ఎనీ వైల్డ్ గెస్ హౌ మచ్ యూ గోనా స్కోర్ ఇన్ దిస్ పేపర్?
So we hope everyone done your exam very well, and let's start our paper discussion.
Okay, before that let me introduce myself. I'm Sneha
And I'm Sheila Dikshit.
So today we are here to discuss about the 10th SSC paper. So before, I asked Sheila ma'am to continue the first sections. Okay.
Right, students. Okay, students, let's discuss about paper in today's exam. I think the paper is very easy and everyone can score maximum marks in our today's paper. Right, students, I will discuss about the session 1 and session 2. Right. Okay, this is the paper today we have done in our public exam. Right, students, we will discuss about session 1 and session 2 while coming to the session 1. First, session 1 anta carries, okay, each question carries 1 mark. Kada? At the first question, let us see about the first question. Name an organism in which the sex is determined by the environmental factors. Right. Okay, let us see about the answer here. For example, the answer turtle. Here, in some reptiles like turtles, okay, alligators, crocodiles, okay, they need the temperature environment they need the temperature in the environment during the critical period. What is the critical period here? During the critical period of egg incubation. And hence it will determine the sex of the offspring. So it is related to the temperature. It is related to the temperature dependent sex determination. సో స్టూడెంట్స్, ఎవరైనా ఆన్సర్ టర్టిల్ లేదా ఎలిగేటర్ లేదా క్రొకోడైల్ పెట్టుంటే యూ డోంట్ నీడ్ టు వరీ. సో ద ఆన్సర్ రిగార్డింగ్ టు దిస్ క్వశ్చన్ ఈస్ ద ఆర్గానిజమ్స్ లైక్ రెప్టైల్స్, లైక్ turtles, crocodiles, alligators, right? They need some temperature in the environment in the critical period for incubation will determine the sex of the organism. So it is the process, or it is the factor which dependent on the temperature to determine the sex. So this is the answer for the first question. While coming to the second one, it is a very simple question, students. Ee question answer entante testosterone. Dash hormone is secreted by testes. So the answer is testosterone. And here testis is the primary male reproductive organ. It is the primary male reproductive organ. Right? And here testis function ఏంటంటే it produces the sperm as well as the male hormone called as testosterone. So testis is the primary male organ. It will produces the sperm as well as the male hormone testosterone. So the answer for this question is testosterone. Students, అందరి క్వశ్చన్ కి ఆన్సర్ అటెంప్ట్ చేశారని నేను అనుకుంటున్నాను. సో దిస్ ఇస్ ద ఆన్సర్ ఫర్ సెకండ్ క్వశ్చన్. While coming to the third question, which part of the brain controls body equilibrium? So the answer is very easy. It is the question from control and coordination, and the answer for this question is cerebellum. Right? Cerebellum. So students, cerebellum anedi ekkada part? It is the part of our back brain. Okay, it is the part of the brain below the cerebellum. Okay, below the cerebrum. సో దీని ఫంక్షన్ ఏంటంటే ఇట్ ఈస్ రెస్పాన్సిబుల్ టు కంట్రోల్ ద వాలంటరీ యాక్షన్స్ ఆఫ్ అవర్ బాడీ. సెరిబ్రమ్ ఫంక్షన్స్ ఏంటి? ఇట్ విల్ కంట్రోల్స్ ద వాలంటరీ యాక్షన్స్. వాలంటరీ యాక్షన్స్ అంటే ఏంటి? ఎవరైనా లైవ్ క్లాసెస్ చూసింటే వాలంటరీ యాక్షన్స్ కోసం నేను ప్రీవియస్గా ఎక్స్ప్లెయిన్ చేశాను. వాలంటరీ యాక్షన్స్ ఆర్ ద యాక్షన్స్ విచ్ ఆర్ అండర్ అవర్ కంట్రోల్, రైట్? ఇట్ విల్ కంట్రోల్స్ ద వాలంటరీ యాక్షన్స్ అండ్ వాలంటరీ మూమెంట్స్ ఆఫ్ అవర్ బాడీ and as well as the balance, posture. Thus it will control the body equilibrium. So the answer for this question is cerebellum. Right students, while coming to the fourth question. Fourth question, what is the chromosomal number in human ovule? Here manaki 4 options icharu. Option A 22 plus Y, option B 22 plus X, option C 44 plus X5, option D 44 plus XX. అయితే ఇందులో ఆన్సర్ ఏంటనేది మనం క్లారిటీ తీసుకొద్దాం. వైల్ కమింగ్ టు 22 plus X is the correct answer here students. ఎలాగంటే let us see. While coming to the human ovum is the haploid cell, right? It is the haploid cell. It means haploid cell ఏంటి? It means it contains half of the chromosomes. It contains half of the chromosomes in somatic cell, right? Half of the chromosomes in somatic cells, that it has total 23 chromosomes, right? Total it has 23 chromosomes, like 22 autosomes and 1 X sex chromosome. So the answer enti? Option B is the correct answer, where 22 plus X is the correct answer. Right students? What is the chromosomal number in human ovule is? So the answer is 22 plus X. So 22 chromosomes are autosomes, and while single X chromosome is the sex chromosome. While coming to the next one, write a slogan on female foeticide. మనం ఫస్ట్ ఫీమేల్ ఫార్టిసైడ్ అంటే ఏంటో తెలుసుకోవాలి. తెలుసుకోకుండా వాటికి ఆన్సర్ అనేది మనం ఎలా రాసిస్తాము? సో వీ హ్యావ్ టు నో ద మీనింగ్ ఆఫ్ ది ఫీమేల్ ఫార్టిసైడ్ హియర్. ఫీమేల్ ఫార్టిసైడ్ ఈజ్ నథింగ్ బట్ it is the practice, right? It is the practice of female aborting. It is the practice of aborting female fetus, right? So దీన్ని బేస్ చేసుకొని మనం ఎలాంటి స్లోగన్స్ని రేస్ చేయొచ్చు? లెట్ us see one example here. I will give one example for you students to stop the foeticide. Okay, female foeticide. Stop female foeticide. Let her see the light. A world without girl child. A world without girl child or children is a world without future. Is a world without future. సో ఇలాంటి స్లోగన్స్ మీ ఓన్గా మీకు ఎలాంటి స్లోగన్ రాయాలనిపిస్తే అవి రాయొచ్చు, బట్ ఇట్ హ్యాస్ టు గివ్ ద సెన్స్ అండ్ మీనింగ్ ఆఫ్ ది స్టాప్ ద ఫీమేల్ ఫార్టిసైడ్. రైట్ స్టూడెంట్స్, ఇలాంటి స్లోగన్స్ మీకు నచ్చిన స్లోగన్ని మీరు రేస్ చేస్తూ ఆన్సర్ ఒక అవేర్నెస్ని క్రియేట్ చేస్తూ మీ స్లోగన్ని మీరు రాయొచ్చు. సో దిస్ ఈస్ ద ఎగ్జాంపుల్ దట్ గివెన్ ఫ్రమ్ మై సైడ్. Stop female foeticide. Let her see the world. A world without girl child or girl children is a world without future. Right. This is about 5th question. While coming to the 6th question, let us see the question here, students. If a trait A exists in 10% of population of an asexually reproducing species and a trait B exists in 60% of the same population, which trait is likely to have arisen early? Right. అయితే ట్రేట్ ఏ అనేది 10% పాపులేషన్లోనే ఎగ్జిస్ట్ అయి ఉంటే ట్రేట్ బి మాత్రం 60% సేమ్ పాపులేషన్లో ఎగ్జిస్ట్ అయింది. అయితే ఇందులో ఏ పాపులేషన్ అనేది త్వరగా ఎరైజన్ అవుతుందని మన క్వశ్చన్ అడిగాడు. సో ద ఆన్సర్ వీ క్యాన్ డిరైవ్ హియర్ ఈజ్ లెట్ us see the most trait B, right? It is existed most percent in population. While coming to trait B, 60% of population లోనే సేమ్ పాపులేషన్లోనే 60% ఇది ఎగ్జిస్ట్ అయింది. సో హియర్ 60% will have most likely it is more likely to arise early. Okay, it is more likely to arise early when we compare to trait A. Right students, let us see how. Because here in asexually reproducing organisms, okay, because in asexually reproducing organisms, while coming to asexually reproducing organisms, the traits that appear earlier, ఏ ట్రేట్ అయితే త్వరగా అపియర్ అవుతుందో, the traits that appear earlier will have more chances to replicate and to spread throughout the portion, okay, throughout the portion of the population. The trait which appears earlier will have more time to replicate and to spread throughout the population. So the answer for this question is trait B. Trait B is the correct answer, and trait B is the thing which arises earlier in the asexual reproducing population. Right students, while coming to the next one. How does ozone formation occur at higher layers of atmosphere? First of all, we have to know అసలు ఓజోన్ లేయర్ అనేది ఎక్కడ ఫామ్ అవుతుంది, ఏ లేయర్లో ఫామ్ అవుతుంది అనేది మనకు తెలియాలి. అప్పుడే ఓజోన్ లేయర్ అనేది ఎలా ఫామ్ అవుతుందో మనకు తెలుస్తుంది. సో ఓజోన్ అనేది ఎక్కడ ఫామ్ అవుతుంది? ఓజోన్ విల్ ఫార్మ్స్ in the stratosphere. Ozone ekkada form avutundi? It will form in the stratosphere, and here the high energy UV radiations will split into oxygen molecules. So it will forms and it will helps in the formation of ozone. So ozone అనేది ఎక్కడ ఫామ్ అవుతుంది? స్ట్రాటోస్పియర్ లేయర్లో. అయితే ఇక్కడ హై ఎనర్జీ యూవీ రేడియేషన్స్ అనేవి ఎలా స్ప్లిట్ అవుతాయంట? ఇట్ విల్ స్ప్లిట్ ఇంటూ ఆక్సిజన్ మాలిక్యూల్స్, హెన్స్ ఇట్ హెల్ప్స్ టు ఫార్మ్ ది ఓజోన్. రైట్ స్టూడెంట్స్, వైల్ కమింగ్ టు ద నెక్స్ట్ వన్. Read the given information and answer the following questions. మనకి ఇక్కడ ఒక టేబుల్ ఇచ్చాడు, ఆ టేబుల్ నుంచి 2 క్వశ్చన్స్ అనేవి మనకు అడిగాడు. While coming to A, shoot of the plant bends and grows towards the sunlight. Which type of response is this? Right students, it is very easy question. చూడండి, మామూలుగా ప్లాంట్స్ అనేవి లైట్ సైడ్ టర్న్ అయితే మనం ఏమంటాము? సన్లైట్ వి కాల్ photo, right? Light is known as photo. And here plant bends and grows towards the sunlight. Which type of response it is showing అని అడిగాడు. సో ఎటువైపు తిరుగుతుంది? It turns towards the light energy. So the type of movement showed here is phototrophism. Right students, the answer for this question is phototrophism. It is A. And while coming to the B, tendrils grow towards the support. Which type of response is this? అయితే ఇక్కడ ఏ టైప్ ఆఫ్ రెస్పాన్స్ చూపిస్తుంది? టెండ్రిల్స్ గ్రోయింగ్ టువర్డ్స్ ద సపోర్ట్ మీన్స్ ఇట్ ఈస్ షోయింగ్ ద థిగ్మోట్రోఫిజం, రైట్? సో ద ఆన్సర్ ఫర్ దిస్ క్వశ్చన్ ఈస్ ఆప్షన్ థిగ్మోట్రోఫిజం. While coming to the next one, observe the flow chart indicating the pathway of air in the human respiratory system. Answer A and B. అయితే ఇక్కడ 2 పార్ట్స్ అనేవి మిస్ అయినాయి మన ఎయిర్ పాత్వేలో, రైట్? పాత్వే ఆఫ్ ఎయిర్లో. ఫస్ట్ ఆఫ్ ఆల్ వీ హ్యావ్ టు నో ద పాత్వే ఆఫ్ ద ఎయిర్, అండ్ దిస్ ఇస్ ద పాత్వే అండ్ దిస్ ఇస్ ద ప్యాసేజ్. ఓకే, ఫస్ట్ పార్ట్ ఈస్ nostrils, right? We already know. And the second one is nasal cavity. And nasal cavity తర్వాత ఏంటి వస్తుంది మనకి స్టూడెంట్స్? బిఫోర్ larynx we will get pharynx, right? And pharynx తర్వాత larynx నుంచి ఎయిర్ ప్యాసేజ్ ఎక్కడికి వెళ్తుంది? It will go to the trachea, and after that it will goes into the bronchi and bronchioles, and after it will get into the lungs and alveoli. Right students, this is the passage and this is the pathway of the air in human respiratory system. అయితే ఇక్కడ missed parts ఏంటి? One is A, it is the pharynx, and one is B, and that is trachea, right? And while coming to the last question in the section 2 is, what precautions do you take to avoid goitering? అయితే మనం ఎలాంటి ప్రికాషన్స్ తీసుకోవాలి ఈ గాయిటరీని అవాయిడ్ చేయాలంటే? ఫస్ట్ ఆఫ్ ఆల్ ద ప్రికాషన్స్ వీ హ్యావ్ టు టేక్ హియర్ ఈజ్ ద ప్రైమరీ ప్రికాషన్. వాట్ ఈస్ ద ప్రైమరీ ప్రికాషన్ వీ హ్యావ్ టు టేక్ హియర్ ఈజ్ ద ప్రైమరీ థింగ్ వీ హ్యావ్ టు ఎన్ష్యూర్ ద adequate intake of iodine, right? The adequate intake of iodine is very, very important. And how we have to take this iodine? Typically in our meals, typically by using iodized salt in our daily meals. So this is one precaution that we can prevent, okay, that we can avoid the goitering. Okay students, we have done session 1 and session 2. Now Sneha ma'am will continue with the session 3 and 4. Thank you all.
సో హలో స్టూడెంట్స్, ఎలా రాశారు? ఇప్పటి వరకు షీలా మ్యామ్ అంతా ఎక్స్ప్లెయిన్ చేశారు కదా. సో హౌ ఇట్స్ ఇట్? వాట్ ఈస్ ద స్కోర్? ఎస్, హాయ్ ప్రసాద్, హాయ్ జోషువా, ఎలా రాశారు? అంటే ఇప్పటి వరకు సొల్యూషన్ చెప్పిన దానిలో ఏమైనా డౌట్స్ ఉన్నాయా? ఓకే, సో ఐ ఎక్స్పెక్ట్ మీరు అందరూ ఇదే రాసి వచ్చారు. ఎవరైనా డిఫరెంట్ ఏమైనా రాసి వచ్చారా ఏంటి? హాయ్ హాయ్ హలో. ఓకే, సో ఐ కెన్ సీ ద స్పిరిట్ హియర్, ఇట్స్ గోయింగ్ హై, రైట్? ఓకే ఓకే, బాగా రాశారు. ఆల్ రైట్. ఓకే, ఫైన్ ఎవ్రీవన్. సో ఎస్, మూవింగ్ ఫార్వర్డ్. So here I would be dealing with the section 3 actually. Okay, so this is for 4 marks. Okay, so the question is, what are the methods used by plants to get rid of waste? Everyone, mirem rasaru. Hello everyone, hello. Yes. Okay, fine. So what are the methods? Yes. All right, fine. So there are various methods through which the plant dispose their waste, right? So one of them is transpiration. What is transpiration? Yes, Transpiration is the loss of excess water from the surface of the leaf through the stomata, right? So, what is this? Excess loss of water in the atmosphere. This is the one method. Then we have diffusion through, diffusion through the stomata and lenticels, right? This is one method. Yes, ఇది రాసారా లేదా? Yes, transpiration is the loss of water. Yes, excess loss of water. What is the second method? We have shedding parts. So the waste is also deposited or accumulated in certain parts of the plants. Like here we have leaf. So leaf will shed off, right? Okay. Fine. Leaves will shed off. Then we have fruits which will drop. Then we have here bark which will peel off, right? Okay, fine. Yes, bark will peel off. Any other methods? Yes, very good. Yes, okay. Apart of this, we have few more like gums and raisins. Okay, though it is useful for us, but it is not useful for the plants. Gums and resins, right? Okay, yes, gums and raisins. Okay, anything else? These are mostly deposited in the non-functional or old xylem. Then we have excretion into soil by roots, right? These are the various methods through which the plant get rid of waste, right? Good afternoon, Divyansh. Yes, okay, fine. So, this is the answer for the 11th one. Everyone, correct Karasachara? Yes, okay, fine. All right, hi Madhu. Okay, fine. Okay, fine. Next question, everyone. So here we have a diagram based question, 4th mark question. So draw a neat label diagram of the human excretory system, and the or one for this question is the B part, which is a draw a neat label diagram of the female reproductive system. Yes, okay. So I'm just drawing something roughly. Okay, here we have kidneys, right? A pair of kidneys. Okay, fine. Okay, here the tubes that carry urine from the kidney and it is deposited or drained in the urinary bladder. Here we have this as ureter. Okay, this is kidney, urinary bladder. Okay, where urine is stored. Then here we have urethra. So this is a rough diagram of the kidneys, right? Okay, fine. All right. Okay, then coming to the female reproductive system. Okay, everyone. So here I am drawing a rough diagram of it. Okay, here we have first. Okay, this is. Okay. Yes, please. So, ide rasara, have you drawn this one? I think, yeah, better than this. But okay, fine. So, here we have uterine fundus. Here, uterine cavity. This is fallopian tube. Okay. This is uterus. Here we have ovary attached to the ligaments. This is ovary. Okay. Here we have cervix, and at the end we have vagina. Right? Here we have cervix. Okay. If you see fallopian tube is further divided into 3 regions, ampulla, isthmic and infundibulum. Here we have fimbriae, right? Clear? Okay, fine. Okay, everyone. So clear, everyone? So moving forward to the next one, the 13th question, suggest few environmental friendly practices to reduce usage of pesticides. Pesticides are what? Pesticides are the chemicals, right? What is the effect of it? If it's exposed for longer period, it may lead to various kinds of pollution, right? So what are the other methods that we can opt for, right? So yes, what you have written? Okay. Yes. All right. So yes, the first one we have the IPM, Integrated Pest Management. Okay, so Integrated Pest Management untadi. So we have prevention and we have chemicals, but this is the last option. Chemicals we use at the end, last option. Ippudu options lenappudu we use chemicals. But here, what is involved in prevention? Here we have monitoring. Okay, monitoring and identification of certain pests. Okay, this is one method. Any other method? Yes, we have various other methods like crop rotation. Then we have intercropping, right? Here we have intercropping. Then soil health management. Okay. Okay, so we have soil health management where we use various compost. Okay, compost, what is the effect of this? Yes, what is compost? Yes, okay. Fine. Then we have various other methods like proper irrigation. Proper irrigation will be a factor also. What is the factor here? Proper irrigation, okay? Proper irrigation leads to avoid overwatering. What will happen if there is overwatering, everyone? Overwatering will lead to high humidity. Okay. Now, if there is high humidity, it will attract various insects and weeds, right? Okay. Then we have certain biological controls. Okay. Everyone, I'm writing here. Okay. We have physical and mechanical methods also. Here we have physical and mechanical methods. So what are the physical and mechanical methods? Here I am talking about weeding. Okay, those crops which compete with the crops. Okay, the waste plants which compete with the crops. Okay, for nutrition, for space, right? We should take it out. This process is called as weeding. Then we have certain traps like sticky traps. Okay, here we have like yellow traps for white flies. We have some other traps also which can be. Okay, so we have some biological controls where we use certain insects and nematodes. Okay, biological control where we use insects like ladybug which feed on aphids. Then we have certain nematodes. These are the methods through which we can reduce the usage of pesticides, everyone. Yes? Clear? Okay. All right. Fine. Yes, ide rasochara, inkemayna rasochara. All right. Fine. All right. Okay, moving forward. So the 14th question, everyone. Write any 4 slogans to create awareness among the people about organ donation. Yes. What are the slogans? So we have so many slogans. Plenty of slogans are there. Okay, so we have like, let's say one we have here, donate organ today for better tomorrow. This is one. Okay. Then we have here like give a life, gift a life. Okay. Then we have organ donation, saves life. Then we have the fourth one like don't wait for miracle, be a miracle. Okay. Okay. Inga chalane unnai, like we have be a hero, be an organ donor, so many. So what did you wrote? Okay. Yes. Meerem rasoccharu. So these are the few examples. You can add few more from your side also. Okay. Fine. All right. So next one, 15th question. This is the section 3, 4th mark question. Plastic is harmful to the environment. Okay. How do you support this statement? Okay. Why plastic is harmful? Because plastic is non-biodegradable. Okay. Non-biodegradable. And it is persistent. Okay. If you see, the plastic remain in the environment like for 100 to 1000 years. Okay. Now how it is affecting, how it is harmful. So here it affects aquatic and terrestrial life forms. Okay. How? Life forms. Okay. How? Okay. See, in aquatic organism, okay, we can see the ingestion of certain chemicals, of certain plastics may lead to the starvation and ultimately death of the organism, right? Then if the aquatic organisms like turtle, fish, if they get tangled, okay, here we can write entangled. If they get entangled in the net or in the waters or anything, it can be anything, right? It will lead to suffocation. This is how it is affecting. This is one method. How in the terrestrial life forms, if you see the landfills, okay, they are accumulated by these plastics. Now this plastic will reduce the fertility of the soil, and this will definitely a harmful way of destroying a terrestrial life forms. Okay, everyone, this is linked to the biomagnification. Okay, biomagnification. Clear? Yes. Yes, everyone. 9-1 explain. Okay, fine. Okay, clear everyone? Okay, fine. Definitely, 9th one will be explained again. Don't worry, Satish. Sheila ma'am will explain again. Don't worry about it. Okay, a part of this, what all we can add here, then this plastic are toxic in nature. They are carcinogenic. Carcinogenic means what? They can cause cancers. This is one reason also. Then microplastic infiltration. Okay, microplastic infiltration. Okay, what is microplastic infiltration? Now you can see tiny particles of the plastics can be found anywhere, be it as drinking water, be it as any human tissue also. Yes. All right. Yes, then it also leads to climate change. How climate change, everyone? Because plastic production and disposal accounts around 3 to 4% or 3 to 5% actually of the global greenhouse gas emission. Okay? Fine? Yes, Prashad. Yes. It leads to cancer. Yes, carcinogenic, those which can cause cancers. Okay, fine. Clear, everyone? Okay, okay. All right. Yes. Anyone has any doubt? Okay, fine. Okay. Moving forward. Okay. So here we have the section 4, 8 marks question. Okay. So always 8 mark question comes in OR. Okay. Here the 16 one. Observe the provided apparatus setup, 2 bell jars with plants and describe the experiment. Okay. This is very famous experiment, everyone. Right? Yeah, okay. And in the OR, we got describe the procedure for followed in your school laboratory to observe the spores in rhizopus. Right? Okay. Okay, Satish. Okay. 8 mark lo monohybrid rayocha. Okay. I'll come back to that, Sandhya. Okay. I'll come back to that. All right. So anyone has any issue? ఇలానే రాసి వచ్చారు కదా. Okay, okay. Fine. Creation? Yes, okay. First question lo doubt unda. Okay. Fine, fine. Okay, okay. Fine, fine. Okay, now here, okay. Don't worry, Satish. Sheila ma'am will explain it again, don't worry. Okay. Fine, fine. Okay, coming back to the 16th question. Observe the provided apparatus setup. Okay, so what are the procedure here? Here we have to take apparatus. What are the apparatus? Here we have bell jars, 2, then potted plants, right? Here we have 2 potted plants, then chemical we have KOH, potassium hydroxide. Then for starch test we have iodine. So but what is this experiment for? What is the aim here? So the aim is the carbon dioxide is necessary for photosynthesis. Okay? Right? Okay. Fine. Okay. Okay, okay, fine. Yes, Sandhya? Okay, fine. So now what happened? We take 2 bell jars. In that we have 2 potted plants. Now what happens? Here in this A setup, we have KOH. Now KOH, what this KOH can do? It can absorb carbon dioxide. Right? Yes or no? Okay, fine. Yes, yes. Okay, now I'll place these 2 plants in an airtight jar. What will happen? Yes, and then I will expose it to the sunlight. So after some times we can observe when we pluck the leaves and do a starch test. The one which is having KOH will not show any iodine positive test. So it will show yellow or brown color. But the one where there was no KOH and it was exposed to sunlight, here we can see starch. How? Because it will show positive iodine test. Okay? Positive iodine test ante it will show blue-black color. Okay? Right? So from here, what we can conclude? We can conclude that carbon dioxide is necessary for photosynthesis. Right? Yes, yes, Sandhya, yes. KOH absorbs carbon dioxide. Right. So clear, everyone? Okay. Okay. Yes. Creation, don't worry. Sheila ma'am, explain chestaru. Okay. Fine. Okay. Here we have this B part. Okay. The B part. Coming to the B part. Okay. I'm just erasing this one. Okay. Fine. Okay. Yeah. Fine. So the second one, describe the procedure you followed in your school laboratory to observe the spores in rhizobus. Yes, rhizobus. Yes, it's bread mold. Very good, Sandhya. Yes, right. Okay, so here what all we require? Apparatus. What are the apparatus required? Yes. What are the apparatus required? So here we require slice of bread, then petri dish, water, then permanent or glass slide. Okay. Okay, just a second. Glass slide. Okay. Then what else do we require? We require compound microscope. Okay. These are the apparatus required for this particular experiment. Okay. Then what is the procedure? Okay. So coming to the procedure, first what we will do? We will grow mold. Okay. How are we gonna grow mold? First we have to moisten it. Moisten it with water. Right? Moisten it with water. Then after that we will leave it in warm dark room, okay, for 2-3 days, right? Now in a closed petri dish. Now what will happen? Okay, after that now we can take this thread and transfer it to a glass slide, okay, cover it. Okay, these threads, when we observe in the microscope, we will see certain thread-like structure. These thread-like structures are called as hyphae. And on the top of it, we have a bob-like structure or a ball-like structure which we call as sporangium. And what is in the sporangium? In the sporangium, we have spores. The spores are tiny dark structures, right? Clear? Okay, fine. Clear everyone? So this was the experiment. So this was the experiment to observe spores in rhizobus. Clear? Okay, fine. Yes, the 17th question everyone. Structure of the nerve cell is suitable for conducting information. Analyze the statement here. Okay. So before that, okay, here we have to draw the diagram. It is very important everyone. Okay, fine. So here, let's try to draw the neuron. Okay. So yes. All right. Okay, fine. Okay. So here, this is a cell body, right? We also call it as cyton or soma. Here we have tiny branched or numerous structures called as dendrite. Okay, so that dendrite are numerous, branched, and branched because it will increase the surface area. More the surface area, more will be the acceptance of the chemicals, right? From the dendrite, the nerve signal is transported to the cell body. Now here on the axon, we have certain cells. These cells are called as Schwann cells. Okay. Now the Schwann cell, what is the function of these Schwann cells? Okay. It will secrete or synthesize a lipid layer. This lipid layer is called as myelin sheath. And myelin sheath is an insulator. It acts as an insulator. Okay. Now what is the function of this? It will cause the jumping action. It will conduct impulse like it is jumping. So we can say it is as saltatory conduction, which prevents the leakage of the nerve impulse, right? Here at the end of the axon, we have a knob-like structure called as axon terminal, in which we have neurotransmitters, right? So what is the flow of the nerve impulse, everyone? So it is from dendrite to cell body, from cell body to the axon. Okay. Now here you need to focus. Okay, just a second. Yes, fine. So what happens? The stimulus reached the dendrite in the form of chemicals from the neurotransmitter. Now here in the axon, in the entire neuron, the nerve impulse is transmitted in the form of electrical signals. When it reaches the axon terminal, it triggers the release of chemicals and it will cross the synapse and it will move to the next neuron. So here we see, okay, fine. Yes, in the neuron we have electrical signals. The nerve impulse is traveled in the form of electrical signals. Okay, it will cross the synapse but in the form of chemical signals, right? So this is the conduction of nerve impulse, right? Okay, fine. All right. Okay, spelling strike of Chasey, wrong Rasan. Okay. Okay, fine. Okay, fine. All right. Okay, so only spelling mistake है क्या? Okay. All right. Okay, traits. Next question. Okay, 17 part B. Traits may be dominant or recessive. How would you analyze this statement as per Mendel's experiment? Which experiment is this? Okay. Madhu, don't worry. Sheila ma'am will explain the first question again. Don't worry about it. Okay. All right. All right. Clear, everyone? Yes. Okay. Now coming to the 17th B part. Okay. I'm just erasing all these things here. Okay. Yes. Everyone, what experiment is this? Yes. Law of dominance, right? Yes, very good, Sandhya. Law of dominance, yes. So if you see here, let's have one monohybrid cross here. Here, let's say we have parents which are pure, tall. Another parent, here we have pure, dwarf. Okay, what is the gamete here? Here we have T, capital T. Here we have small t, right? Yes or no? Okay. All right. Fine. Okay. So when we cross, what is the F1 generation here? So here we have capital T, small t, which is what? It is a hybrid, but it is a tall, right? So what law of dominance says? When an inherited pair of allele is heterozygous, the one which expresses itself is a dominant one and the one which could not express is the recessive one, right? So if we see the F2 generation here, so we get after selfing, right? So what is the generation here? We have capital T. Okay, I'm hiding it. Okay, fine. I'm writing here, everyone. Okay. So what is the F2 generation? This is capital T, capital T, small t, capital T, small t, and small t. If you see here, 75% of it are tall and the 25% is the dwarf one, right? Yes, very good, Sandhya. Yes, 1:2:1. Yes, this is what? Yes, very good. Genotypic ratio and the phenotypic ratio is? Yes, very good. Yes, 3:1. Very good, Sandhya. So, yes, Sandhya. Yes, Satish. Okay. So, clear, everyone? Ma'am, first question answers Neil. Okay. So, we have so many doubts. Okay. Fine. Fine, all right, okay. So yes, I hope the paper went very well. So what is your score, everyone? Okay. 50 నుంచి ఎంత ఎక్స్పెక్ట్ చేయొచ్చు? Okay, fine. Okay. All right, yeah. I can see there are a lot of doubts in the section 1. Yes, okay. Satish, Madhu, okay. Sandhya. All right. Okay. So, okay, over to Sheila ma'am. Okay. Sheila ma'am will explain the section 1. All right, everyone. Okay.
Right, Madhu, you have a doubt. Like from the first question, right? You are asking that you wrote in the snail answer. And the snails are not reptiles. I gave you a reply in the chat box also. I think you did not see the answer. While coming to the answer, see here, Madhu, turtles, right? We can give some answers like, for example, I will write here in the end slide. Turtles or alligators or crocodiles. వీటిలో నువ్వు ఆన్సర్ ఏం చేసినా సరే నీకు మార్క్స్ వస్తాయి బికాస్ దే ఆర్ రెప్టైల్స్. ఎందుకంటే రెప్టైల్స్ మాత్రం ఒక క్రిటికల్ పీరియడ్ ఉంటుంది. ఆ క్రిటికల్ పీరియడ్ ఏంటంటే ఇన్క్యుబేషన్ ఆఫ్ ది ఎగ్. దే నీడ్ సర్టెన్ టెంపరేచర్ ఫ్రమ్ ది ఎన్విరాన్మెంట్. ఆ సర్టెన్ పీరియడ్లోని ఆ క్రిటికల్ టైంలోని ఇవేం చేసుకుంటాయంటే ఇన్క్యుబేషన్ టైంలోని ఎన్విరాన్మెంట్లో ఉన్న టెంపరేచర్ని యూజ్ చేసుకొని ఆఫ్ స్ప్రింగ్ యొక్క సెక్స్ని డిటర్మైన్ చేస్తాయి, రైట్? సో ఇట్ ఈస్ డిటర్మైన్డ్ ఆర్ ఇట్ ఈస్ ఎ డిపెండెంట్ సెక్స్, ఓకే, సెక్స్ డిపెండెంట్ ప్రాసెస్ విచ్ డిపెండ్స్ ఆన్ టెంపరేచర్, రైట్? ఈ టెంపరేచర్ని బేస్ చేసుకొని ఆఫ్ స్ప్రింగ్ యొక్క సెక్స్ని డిటర్మైన్ చేస్తాయి. కాబట్టి దీని ఆన్సర్ మనం టర్టిల్స్, రెప్టైల్స్, ఓకే, రెప్టైల్స్ ఆర్గానిజమ్స్ లైక్ ఎలిగేటర్స్, క్రొకడైల్స్ అని మనం పెట్టొచ్చు. సో స్నేల్స్ అనేది కరెక్ట్ ఆన్సర్ కాదు. Any more doubts, students? ఓకే స్టూడెంట్స్, ఇంకెవరికి ఏమైనా డౌట్స్ ఉన్నాయా? సెక్షన్ 1 నుంచి సెక్షన్ 2 నుంచి ఏమైనా డౌట్స్ ఉంటే వాటిని మనం సాల్వ్ చేద్దాం. 4th క్వశ్చన్, రైట్. వైల్ కమింగ్ టు ద 4th క్వశ్చన్ ఆప్షన్ బి ఇస్ ది కరెక్ట్ ఆన్సర్, 22 + x. ఎందుకంటే మన హ్యూమన్ బాడీలో క్రోమోజోమ్స్ మొత్తం ఎన్ని ఉంటాయి, రైట్? ఆప్షన్ బి ఇస్ ద కరెక్ట్ ఆన్సర్, 22 + x. ఓకే, 1 క్వశ్చన్ ఫ్రమ్ సెషన్ 2 ఎట్ లాస్ట్. ఎస్, నువ్వు ఆప్షనల్ అని పెట్టేసి లేదంటే ఎక్స్ట్రా అని రోమన్ నెంబర్, ఓకే, సెషన్ 1 రోమన్ నెంబర్, క్వశ్చన్ నెంబర్ వేసి నీ ఆన్సర్ నువ్వు అటెంప్ట్ చేస్తే ఖచ్చితంగా నీకు మార్క్స్ వస్తాయి. సో డోంట్ వరీ. ఓకే రిత్విక్, అండ్ వైల్ 8th క్వశ్చన్. ఓకే స్టూడెంట్స్, వైల్ కమింగ్ టు ద 8th క్వశ్చన్, ఓకే వెయిట్. ఓకే, మనకి 8th క్వశ్చన్ ఏంటి ఇక్కడ? వైల్ కమింగ్ టు ద 8th క్వశ్చన్, రీడ్ ద గివెన్ ఇన్ఫర్మేషన్ అండ్ ఆన్సర్ ద ఫాలోయింగ్ క్వశ్చన్స్ అన్నారు. సో ఒక టేబుల్ ఇచ్చి దాని నుంచి 2 క్వశ్చన్స్ అనేవి మనకు అడిగారు. లైక్ ఏంటి? షూట్ ఆఫ్ ద ప్లాంట్ బెండ్స్ అండ్ గ్రో టువర్డ్స్ ది సన్లైట్. విచ్ టైప్ ఆఫ్ రెస్పాన్స్ ఈస్ దిస్? అయితే ఇక్కడ ప్లాంట్ అనేది ఇట్ ఈస్ బెండింగ్ టువర్డ్స్ ద సన్లైట్ అండ్ ఇట్ ఈస్ గ్రోయింగ్ టువర్డ్స్ ద సన్లైట్ అని అన్నాడు. సో ద ఆన్సర్ ఫర్ దిస్ క్వశ్చన్ ఈస్ ఫోటోట్రోఫిజం ఈస్ ద కరెక్ట్ ఆన్సర్ ఫర్ ఆప్షన్ ఏ. బికాస్ the organism, okay, the plant which shows to bending towards and growing towards the sunlight is phototrophism. Here photo means light. ఓకే స్టూడెంట్స్, సో ఆన్సర్ ఆప్షన్ ఏ. ఫర్ ఆప్షన్ ఏ ఆన్సర్ ఏంటంటే ఫోటోట్రోఫిజం. వైల్ కమింగ్ టు ద బి, టెండ్రిల్స్ గ్రో టువర్డ్స్ ది సపోర్ట్, విచ్ టైప్ ఆఫ్ రెస్పాన్స్ ఈస్ దిస్ అన్నాడు. సో ఆప్షన్ థిగ్మోట్రోఫిజం. ఓకే, టచ్ స్టిమ్యులా, ఇక్కడ చూపించే స్టిమ్యులస్ ఏంటి, టచ్. సో ద ఆన్సర్ ఈస్ ద tropical movement here, thigmotrophism for B. Okay students, 8th question is this. And some asked about the 4th question. I think 4th question answer ఏంటంటే ఆప్షన్ బి ఇస్ ది కరెక్ట్ హియర్. ఎందుకంటే ఓవం అనేది ఇస్ ఎ హ్యాప్లాయిడ్ సెల్. ఓకే, ఇట్ ఈస్ ఎన్ హ్యాప్లాయిడ్ సెల్. హ్యాప్లాయిడ్ సెల్ ఈస్ నథింగ్ బట్ ఇట్ ఈస్ ఇన్ ద రైట్. Okay, right, it is in the, it means right, the chromosomes wherein the here the number of chromosomes of a somatic cell that present, how many chromosomes we have, 23, right? అంటే టోటల్ క్రోమోజోమ్స్ 22 ఉంటే అందులో 22 క్రోమోజోమ్స్ ఏంటి, ఆటోజోమ్స్, అండ్ ద రిమైనింగ్ 1 క్రోమోజోమ్, 1 X క్రోమోజోమ్ ఈజ్ సెక్స్ క్రోమోజోమ్. సో ద ఆన్సర్ ఈజ్ ఆప్షన్ బి, 22 ప్లస్ X ఈజ్ ది కరెక్ట్ ఆన్సర్, రైట్. క్వశ్చన్ నెంబర్ తప్పేసి ఆన్సర్ రాస్తే మనకి ఆ ఆప్షన్ ఏంటి, దానికి మార్క్స్ అనేది ఇవ్వరు. ఓకే రిత్విక్, మనకి ఆన్సర్ అనేది కరెక్ట్ అవ్వాలంటే మనం ఎంత కరెక్ట్గా ఆన్సర్ రాసినా క్వశ్చన్ నెంబర్ అండ్ రోమన్ నెంబర్, సెక్షన్ నెంబర్, ఆప్షన్ అక్కడ మెన్షన్ చేయకుండా ఆన్సర్ రాసినా మన మార్క్స్ అనేవి వచ్చే ఛాన్సెస్ చాలా తక్కువ, రైట్. వీ హ్యావ్ టు బి వెరీ వెరీ క్లియర్ వెన్ వీఆర్ రైటింగ్ ద ఆన్సర్, ఇంక్లూడింగ్ విత్ క్వశ్చన్ నెంబర్ ఆల్సో, ఇంక్లూడింగ్ విత్ సెషన్ నెంబర్ ఆల్సో. ఓకే స్టూడెంట్స్, ఎనీ మోర్ డౌట్స్ ఫ్రమ్ సెషన్ 1 and సెషన్ 2? Right. Okay students, I hope everyone done your exam very very well. పేపర్ కూడా చాలా ఈజీగా వచ్చింది అండ్ ఐ హోపింగ్ బెస్ట్ ఫర్ యూ ఆల్. మీరు మ్యాక్సిమం మార్క్స్ని స్కోర్ చేస్తారు, ఎవరు కూడా డిసప్పాయింట్ అవ్వకండి. అండ్ బీ ఫ్రమ్ టుడే ఆన్వర్డ్స్ ఫ్రమ్ యువర్ బయాలజీ ఎగ్జామ్, రైట్ స్టూడెంట్స్. థ్యాంక్ యూ ఆల్, సీ యూ సూన్ అండ్ డోంట్ ఫర్గెట్ టు ఫాలో అవర్ క్లాస్, SISYA class, and do subscribe. Thank you all.